talking about DNA and DNA structure. DNA stands for deoxyribonucleic acid, D for deoxyribo, N for nucleic, A for acid. DNA is found in the chromosomes in the nucleus, and it's also found in the mitochondria and chloroplast of cells. DNA is a macromolecule. Macro means big, and so DNA is a big molecule. DNA is negatively charged because of the phosphate groups that are found in its backbone. DNA is made up of nucleotides instead of nucleosides. The difference between the two is that nucleotides contains a nitrogenous base, ribose sugars, and phosphate groups. In DNA, the ribose sugar is a deoxyribose sugar, meaning it's lacking in oxygen. Now, as opposed to nucleosides, which only contains nitrogenous bases and ribose sugar. Now, the difference between RNA and DNA is that DNA, the ribose sugar, like I said, is a deoxyribose sugar as opposed to RNA, in which the ribose sugar is a ribose sugar. And the reason DNA sugar is called a deoxyribose sugar is because it's lacking in oxygen. It's lacking in oxygen right here. That's why it's called deoxy. Oxy for oxygen. Now the nitrogenous bases are called nitrogenous bases because they contain nitrogens in the ring. Now there are 5 types of nitrogenous bases that are found in DNA and RNA. Only 4 of them are found in DNA, and only 4 of them are found in RNA. The first type of nitrogenous base is called adenine, and then you have guanine, and then you have cytosine, uracil, and thymine. In DNA, you will only find adenine, guanine, cytosine, and thymine. And in RNA, you will only find adenine, guanine, cytosine, and uracil. And we'll talk more about that in a little bit. The nitrogenous bases are further classified into 2 groups, one called purines and the other pyrimidines. The purines are adenine and the guanine bases. When you look at their structure, adenine and guanine structure, you see 2 rings. So you can think of wedding rings when you see their structure, when you think of adenine and guanine. The hexagon, right? You can think of that being the part of the ring where you put your finger through. And the pentagon would be the diamond of the ring, where the diamond is. And so you can think of wedding rings when you think of adenine and guanine. And you can use the expression pure as gold to remember that adenine and guanine are purines, pure for purines, the A and as for adenine, and the G in gold for guanine. Then you have the pyrimidines. The pyrimidines are cytosine, uracil, and thymine. They all only have 1 ring in their structure. And so you can use the expression cut the pie. Cut, C for cytosine, U for uracil, and T for thymine. Pie for pyrimidines, so cut the pie is the expression. Now you need to know the difference in structures for the MCAT. So the way I remember it is by looking at the carbonyl groups in all of their structures. So adenine has no carbonyl groups. Guanine has a carbonyl group right here, right? This is a carbonyl group right here. So guanine has a carbonyl group. Adenine has 0 carbonyl group. Cytosine also has a carbonyl group, but it's just a 1 ring structure as opposed to guanine, which has 2 rings in the structure. Now, for you to go to cytosine to uracil, you just add another carbonyl group, right? There's 2 carbonyl groups in uracil. Now, to go from uracil to thymine, you add a methyl group. So you have the 2 carbonyl groups plus a methyl group. So from adenine to guanine, that's not the only difference, but for me to remember, that's how I try to remember the structure. So from adenine to guanine, you add a carbonyl group and other things. You add other things too. You do some modification, but just to remember There's a carbonyl group in guanine as opposed to adenine. And cytosine also has a carbonyl group, but it only contains one ring in its structure. Uracil, for uracil, you have 2 carbonyl groups. And for thymine, you have 2 carbonyl groups plus a methyl group for thymine. Now, going back to DNA and RNA, DNA is double-stranded, right? And it only contains the bases adenine, guanine, cytosine, thymine, A, G, C, T. And these bases are paired with each other because DNA is double-stranded. And so adenine, A, the adenine bases are always paired to the thymine bases. And they're paired using 2 hydrogen bonds. Now for guanine and cytosine, they're also paired with each other. And they're paired using 3 hydrogen bonds. And so their bond is so much stronger than adenine and thymine. Now, uracil is not found in DNA. Uracil is found in RNA. And also, thymine is not found in RNA. Thymine is only found in DNA. And the reason why uracil is not found in DNA is because when cytosine is degraded, it converts into uracil. And that's a problem because if you find uracil, if uracil was, found in DNA and you see a uracil, you wouldn't know if it's an original uracil or if it's a degraded cytosine. And so to just get rid of this whole confusion, instead of uracil, you have thymine. And so there's no uracil in DNA. Only adenine, guanine, cytosine, and thymine. But like I said, there are uracil in RNA, and there are no thymine in RNA. Now there's more about DNA in the next video. So see you then.